Thank you for that wonderful introduction, Jeanette. And I also want to thank the organizers for inviting me, and of course the funding agencies without which this work would not have been possible. I want to first give you a brief introduction to Parkinson's disease, which is one of the pathologies that's also really motivating this work. What this is showing is a caricature of someone with Parkinson's disease who has a tremor. Their body is a bit rigid or with a stooped posture, and they take short, shuffling steps. And this is Michael J. Fox, who is a famous child television actor and is one of the people who has Parkinson's and has set up a foundation to fund more research into Parkinson's, as many of you know. So we actually know quite a bit about the cause of Parkinson's disease, and this is showing the cortex, but within that is the basal ganglia, which consists of the caudate and proteomic collections of cells, nuclei, sometimes called ganglia, right here. And also shown here, the motor cortex, all parts of it project to the basal ganglia. This is a striatum, which collectively refers to the caudate and the proteomic, and it projects to the globus pallidus and then to the thalamus. So we have these feedback loops controlling movement. And of course, another very important structure is the substantia nigra. Now, the substantia nigra is called that because it shows up black when you use certain stains. And this is also showing where it's located in an MRI. It's actually part of the midbrain, but the critical feature of this is that it produces dopamine. And in Parkinson's disease, the substantia nigra dies, and so instead of having dopamine released onto striatal neurons producing normal movement, instead we have little to no dopamine released, and then we get movement disorders. Well, what is the dopamine doing? Well, it's binding to these metabotropic or G protein coupled receptors to activate intracellular signaling cascades. And of course, the dopamine is important not just for Parkinson's, as we heard yesterday, it's important for many other types of behavior, such as habits. We all, I'm sure, have found ourselves in a situation where we're driving to a particular destination, and in fact, we might end up doing the wrong turn because we stopped thinking about it and we took the turn that we usually take. Skill learning also requires a striatum, and this is showing a picture of Chico Marx playing the piano, and this is a very characteristic glissando that he always does. And then reward learning. Typically we're studying this in rats or monkeys, where we're trying to increase the behavior of an animal by giving it a reward. And all of these are impaired with dopamine loss. And we heard yesterday, it was really nice that I'm following these wonderful talks on the role of dopamine, but rewards actually cause these dopamine neurons to fire. So when we talk about dopamine, we're not referring to just the tonic dopamine, but a dynamically modulated dopamine. So this is showing that neurons in the substantia nigra or the ventral tegmentum will fire in response to reward. But these neurons will also learn to fire in response to a cue that predicts a reward. And the neurons will also fire in response to novelty. Well, that firing indeed causes an increase in the dopamine, here shown in Stephanie Craig's lab, using measuring the dopamine using dialysis. And yet another disease which involves dopamine is addiction. So all of the drugs of abuse enhance dopamine release. And it's thought that part of the problems with drug abuse is that the rewarding effects of these drugs hijack the normal mechanisms for reward learning. And they're so strong that now these drug addicts are not able to properly respond to normal rewards. Well, we heard yesterday more about network level effects, how dopamine will control D1 versus D2 neurons differently, and it does that by intracellular cascades, and that's really what the focus of this talk is. So this is showing, of course, the cortex again, actually projects to spiny projection neurons. So this is a picture of a spiny projection neuron. This is a schematic showing the cortical inputs, and these neurons are located in the striatum. And the dopamine is also projecting to these neurons. So this is showing a tyrosine hydroxylase stain of dopamine neurons and I'm showing the projection through these spiny projection neurons, which again constitute about 95% of the neurons in the striatum. And the hypothesis is that the dopamine is causing synaptic plasticity or plasticity of ionic channels in these neurons, which is responsible for learning by the whole organism. So we think that this might be happening. Sensory inputs or movements, such as this would be a picture of my dog, is encoded as a pattern of cortical inputs, which are, providing inputs to various places on the dendritic tree of the spiny projection neuron. And then reward, and that's shown here by chocolate. And don't worry, I'm not feeding chocolate to the dog. I'm feeding myself chocolate after I look at the dog. And that's going to activate the dopamine neurons, and that's going to cause learning in these neurons. So one of the big questions is, how does the conjunction of cortical glutamatergic input and dopaminergic inputs cause learning? And I can put that a different way. In fact, I need to phrase that question a little bit more precisely to answer it with a model. And so here are my 3 different cortical inputs. And this cortical input fires consistently with presentation of some pattern. And these other cortical inputs perhaps represent something in the environment or the internal state of the animal, so they fire inconsistently with these pattern presentations. And then the reward, or the dopamine neurons, fire consistently after the cortical inputs. And the question is, how does this neuron know that it should be strengthening these synapses, or this synapse, and not these synapses? And even more of a puzzle is that we're rewarding things that came before the dopamine, not things that came after. Animals only reinforce the actions that came before the reward. Well, the answer to that must be in these networks of signaling plasticity, which control synaptic plasticity. And one common approach for trying to understand this learning is using genomics and proteomics and then graph theory. So you have a lot of these networks of molecules. This shows how they differ slightly between the D2 and the D1 neurons. And we can try and analyze these networks. But the problem with these approaches is that it ignores space and time for the most part. These approaches don't distinguish between molecules in the spine or the dendrite or the soma or the axon, and these techniques don't evaluate how these different signaling pathways may be active differentially depending on the temporal pattern. Here's an example showing that they also can't address questions of spatial specificity, which is very important. And we know this is the work from 2 different labs. This one by Lee et al. Is imaging CAM kinase. And what they're doing is they're uncaging glutamate onto this spine right here, shown in blue here. It's not active, and they're looking at the CAM kinase activity. And they don't see CAM kinase activity as much in an adjacent spine. And so this is the temporal pattern. Here's this nice CAM kinase response, but it's very small in adjacent spine. And it's not CAM kinase alone that shows some degree of spatial specificity. In this image from Carl Svoboda's lab, he's looking at RAS activation, and again he's looking at 2 different spines, and you can see that this spine shows a very large activation, this spine shows a much smaller one, again here quantified over time. So we want to do some simulations that can address the question of what are the mechanisms producing spatial specificity. And this is a problem, as in it's a difficult simulation to do, because on the one hand we have these spines, and calcium and glutamate receptors are located on the spines, and when calcium flows in, it pretty much stays confined to the spine, but in such a small volume, 60 molecules will produce a 1 micromolar concentration. So we're talking about very few molecules, and therefore these reactions occur stochastically. But we have other molecules, perhaps our dopamine receptors are located on the dendrite, or at the base of the spine, and they produce cyclic AMP, and for the cyclic AMP to diffuse or to interact with the calcium requires diffusion. And we have large numbers of molecules in the spine. The cyclic AMP concentration would be pretty high, and its ATP precursor is even higher. So our problem is that we have very large numbers of molecules, and yet we have to model these things stochastically. Well, there are many different, well, many is perhaps an exaggeration, but there are several different types of stochastic simulators that are out there. And we heard a really nice talk yesterday morning which used MCell to do some simulations, spatial stochastic simulations. And a couple others that have been used in the neuroscience community are Smalden and CDS. And in these simulators, individual molecules are represented as point-based particles. They can diffuse a random distance and a random direction. So here's different types of diffusion, or different directions and distances of diffusion with each time step. Or if two reacting molecules pass close by, they can react, and it can be reversible. The problems, or the limitations, I should say, it's not a problem, but the limitations of these simulators is that the number of computations increases with the number of molecules. So though you can simulate large spaces, the volume that you simulate actually is not so important. The total number of molecules dictates how long your simulation will be, which means that I can't tell you what size simulation you can do, because of course it depends on the hardware, which is improving every day, but it still makes very large simulations difficult. So another very common algorithm, especially in the computational cell biology community, was proposed by Gillespie. Now, the basic Gillespie algorithm is a non-spatial algorithm. The algorithm works like this. You calculate, for each reaction, you calculate a propensity, which is the rate constant, times the number of molecules. And if it's a bimolecular reaction, then you multiply it by the other reactant. And we call that, and then you calculate the propensity that any reaction will occur, which is just summing up all the propensities. And then you choose two random numbers, R1, and the log of 1 over R1 divided by the total propensities is the time when the next reaction will occur. And this next reaction time has an exponential distribution. Well, now you know when the reaction will occur, but which reaction will occur? And so you use the second random number to calculate which reaction will occur. And so you do that, and then you recalculate everything. And so you move through this trajectory. And it's called the exact algorithm because he's shown that theoretically it will give you an exact representation of one trajectory through the space. This algorithm is used quite a lot. One of the problems with this also is that the computations will increase with the number of molecules because these values go up and therefore this time step goes down. But of course this is not a spatial algorithm. However, there have been several types of extensions to the Gillespie algorithm. One is to allow it to do spatial simulations. And so the spatial Gillespie, you take your morphology and you subdivide it into small compartments, shown here some radial compartments, and then you'll add the propensity of diffusion. So diffusion is treated as, say, another reaction, except its propensity now is calculated from the diffusion constant. And then you just implement the typical Gillespie algorithm. Gillespie himself also developed another extension to his algorithm called tau leap. Now, he developed a non-spatial tau leap, and the idea was to make an approximation of the exact algorithm, which allowed you to simulate more efficiently. And so you allow multiple reaction events to occur at each time step, and the time step is called tau, hence you're leaping down the time space. And so given your time step tau, you then for each reaction J, you calculate how many events are occurring. For example, how many calcium molecules are binding to that N terminal of the calmodulin. So my lab was, I was inspired by this tau leap because I figured that if I wanted to do a large volume, for example, an entire dendritic tree and all the reactions in there, I needed to do some approximation, but I still needed a stochastic method. And so my lab developed a spatial extension to Gillespie's tau leap algorithm. And so just like tau leap will allow multiple reaction events during each time step, NeuroRD will allow multiple reaction events and diffusion events to occur each time step. We still have to subdivide the morphology into compartments shown here, and then in order to further increase efficiency, we're going to pre-calculate our binomial and Poisson random numbers and store them in tables. And now at each time step, given the probability that one reaction event will occur, we can look up in this table the probability that, say, 2 or 3 of these molecules will diffuse out of the compartment, or that perhaps a couple of these molecules will react with this other molecule. So, well, we have to show that it's accurate. And as I said, it is an approximation, so we need to know how accurate it is. We might get the same means, but not the same variances. And what this is comparing just a very simple reaction and diffusion. So our distance, this is, I think, a 10 by 10 micron structure, and I have a couple of molecules which can react. And the black and gray lines are the deterministic reaction, and you can see that the stochastic reactions surround the deterministic, and that the red represents Smalden and the blue represents NeuroRD, and they pretty well overlap. And the other thing you should see when we look at molecule B, notice this goes from 0 to 10 molecules, is that the deterministic reaction shows quite a difference, sort of this sigmoid curve when it's at a further distance from the source, but you really can't see that with the stochastic. And so this is why we need to do stochastic reactions. We're not really going to get these differences. And NeuroRD is up to 60 times faster than Smalden, and that number 60 comes from these simulations we did. Clearly, the improvement in speed depends on the simulation. But the most notable one is these 2 rows of the table. The only difference between these is the total number of molecules. For this second simulation, it's 10 times the number of the first simulation. And Smalden goes from taking almost 3 minutes to 2 hours and 20 minutes. And with NeuroRD, it goes from 5 seconds to 8 seconds. And it's not the 5 seconds versus 2 minutes, which is significant. It's the increase in computation time as you increase number of molecules. And this number of molecules, by the way, is not some ridiculously high number. It's within the physiological range. So the disadvantage of NeuroRD is that computation time does indeed increase with the number of compartments, but it does not significantly increase with the number of molecules. So if you had a large volume which was sparsely populated, this would not be the software to use. But if you had a smaller volume or that had high concentrations of molecules, then this software would be more computationally efficient. But again, it is an approximation, so depending on the question you are asking, you have to decide, is this appropriate or something else is appropriate? And we're continuing to develop NeuroRD because we still want larger volumes. I cannot yet do an entire dendrite. One of the approaches is to partition what's called critical and non-critical reactions in diffusion. Gillespie has further developed his tau leap algorithm, and the critical reactions are those with few numbers of molecules, the non-critical ones where there are large numbers of molecules, and you would use tau leap for the non-critical, and then the exact algorithm for the critical molecules, and we've implemented that in our spatial approach. And this shows the space average mean using a representative set of reactions, and this shows you that the number of time steps needed to do the simulation goes down dramatically. We've also added diffusion based on the gradient, and interestingly, though the mean is very similar, we actually have, not surprisingly, much smaller variances. And so we're still exploring where you could afford to have that reduction in variance and where you can't afford to have that reduction in variance. But now I want to explain how we use this now to address some spatial and temporal aspects of synaptic plasticity. And I'm going to start with an experimental result from my lab, which is that pKa location is important. And what I mean by location, I mean subcellular location. So this first experiment is showing high-frequency induced synaptic plasticity in the striatum, and that if we add the pKa inhibitor peptide into the pipette, we block our synaptic plasticity. So this is a pKa-dependent synaptic plasticity. But we're now going to do an experiment where we, instead of adding pKa inhibitor, we're going to add something called HT31 peptide. This is a fragment of the anchoring protein. So there are anchoring proteins, or structural proteins, which bind to pKa and hold it immobilized, relatively immobilized, in various parts of the cell, within the cell. And the HT31 peptide, by binding to this pKa binding site, prevents the pKa from anchoring. So maybe it's floating around in the cytoplasm. And what we show right here is that when you use the active peptide, but not the inactive peptide, you block LTP in the striatum. So we want to know why pKa needs to be anchored. So one possibility is that pKa needs to be near the source of molecules that activate it. And another possibility is that it doesn't need to be near the source, but it needs to be near its target molecules, such as the glutamate receptor. So what if the dopamine receptor and the adenylate cyclase were located here, and the cyclic AMP was produced, and cyclic AMP has a relatively low molecular weight, so it can diffuse around, and if the protein kinase A is anchored in the spine, the cyclic AMP might find it, bind to it, activate the pKa, and now when pKa gets active, the catalytic subunit dissociates from the regulatory subunit, and now it's free to diffuse also, but it has a larger molecular weight, therefore its diffusion constant is lower. So maybe the pKa needs to be near its glutamate receptor because it's not going to diffuse very far. Well, if that's the case, then we might expect also to see quite a strong degree of spatial specificity. So one possibility also is that anchoring helps produce spatial specificity. So to address these questions, we need to set up a model of signaling pathways within a dendrite with spines. And this just shows the initial set of signaling pathways that we implemented. The dopamine receptor, which activates adenylate cyclase, which then produces cyclic AMP and binds to pKa. We also have calcium influx through the NMDA receptor, and calcium has several targets. pKa has several targets, including the AMPA receptor, which is clearly at the PSD, but also DARK32 and a few other molecules. We also have CAM kinase in this model, and I won't go more into these signaling pathways because it would be a bit overwhelming at this point. But for all of these reactions, we need to get the kinetics, and we typically go into the literature. There are some databases. This one in particular, developed by Rupi Bala, who is actually here in the audience, is a really nice resource. But you have to be careful about these databases. For example, this one, I just went and put in cyclic AMP, and it didn't find any reactions, probably because I don't know how to use it. But many of these reaction rates come from, say, bacteria, or it's at a different pH or at a different temperature. So the neuroinformatics type of databases that we've been hearing about, they're not as developed in this area, in part because kinetics is just very difficult. And you don't have to believe me, you can talk to a biochemist about that. So now we take all of our reactions and we put them into the morphology. And what we're starting with is this 8 micron long piece of dendrite, and we have 4 spines here. And we've just meshed this in 2 dimensions for now, and we have a submembrane domain, which means that we have some of our molecules only located in the submembrane region, and other molecules are free to diffuse or might actually be anchored in the cytosol, which means that they're just immobilized, but they're everywhere. And of course, our AMPA receptor is only at the PSD. And here's our experimental design. What we want to know is, why is pKa anchored? Does it need to be anchored near the dopamine receptor and the adenylate cyclase producing cyclic AMP, as it is in this case and this case? Or does it need to be anchored near its target, such as the glutamate receptor? And that's shown here in this case, and in this case, where PKA is anchored in the spine. And we just did this 2x2 design. And our results show that, contrary to what I had predicted initially, protein kinase A needs to be anchored near the cyclase. So these two cases show when PKA are anchored near the cyclase. In this case, they're both in the spine. In this case, they're in the dendrite. And in these cases, the PKA is not anchored near the cyclase, but in this brown case, this one, the PKA actually is in the spine near the glutamate receptor. Now this is not as noisy because this is dark 32, there's a lot of it, but what I'm really interested in is the GluR1, and we see the same pattern. So this molecule is everywhere, so you could imagine that being anchored in the spine wouldn't help. The GluR1 is only in the spine, and we see the same pattern. Okay, well there are other really compelling questions about space, especially in the striatum. And so one of these issues is whether dopamine synaptic transmission is more similar to typical synaptic transmission, and that the release site needs to be close to the receptors, or whether dopamine works by volume transmission. There's some idea that the dopamine, because the terminals are small and pretty much scattered throughout the neuropil, and the dopamine receptors appear to be everywhere, although a lot of people don't believe that the antibodies are clean enough. But nonetheless, there's this idea that only the temporal pattern of dopamine matters, but then you have other people who think that the spatial pattern does matter, and that's shown here. This is a nice simulation which is showing that you do indeed have these high concentration regions. And so it's not just one uniform concentration gradient. We were working at the time off of this data, so what we did was we adjusted our dopamine diffusion constant to allow the release of dopamine in one place, and then as it diffused to other places, the concentration would be about 50% as high. And when we did this simulation, this is our control cyclic AMP and PKA activity measured by two different targets. And when we had a gradient, you can indeed see a decrease in the concentration of these molecules, but it's not very large. So from this, we're not really convinced that the dopamine gradient, which might exist, is all that significant. But we were using a small piece of dendrite, so now let's use a larger dendrite with many spines shown here. We're going to release the dopamine here, and we're going to say, well, would we, if the dopamine signaling had even more spatial specificity, as shown in the Dreyer study, would we see spatial specificity in the signaling molecules inside the neuron? Because that's really what matters. So our cyclic AMP has a very nice spatial gradient. This is where we're releasing the dopamine, and you can see that about 10 microns away, we don't really see much cyclic AMP. And this is just a spatial average, or we're averaging over a small period of time, and you can see this mono-exponential decay. So we clearly see a gradient in cyclic AMP under these conditions. But we do not see spatial specificity in PKA activity. Now, I'm not showing you the PKA because the signal is very noisy. So what I'm looking at, again, are the dark 32 targets, and you might see the hint of a gradient here, but it's not very prominent. And then phosphorylated GluR1, again, no spatial gradient. Well, why are we not seeing a spatial gradient in PKA? Well, one possibility is that dark 32 can diffuse, and the phosphorylated form can diffuse, at least in our model. So maybe that's the problem. Maybe PKA would have a spatial gradient. So we removed the diffusion of dark, and indeed we see this nice spatial gradient. Well, then we said, well, is it really the diffusion of the phosphorylated form, or is dark 32 acting like a calcium buffer? Calcium buffers bind calcium to sort of temporarily inactivate it, but the calcium buffers themselves will diffuse, and we all know that, with all the calcium imaging experiments that are done, that those Fura-2s and Oregon green, they help the calcium move from a high concentration region to a low concentration region. So we hypothesized that that's what the dark 32 was doing. When the pKa bound to the dark 32, it was now the diffusion was being facilitated. So we just eliminated diffusion of those two forms, and we still got the nice gradient. So it's not the diffusion of the phosphorylated form. And the last thing we did was to say, well, why is there's another dark form. When it's phosphorylated on threonine 75, it actually binds to and inhibits pKa. So that's an inactivation mechanism, because there's a lot of evidence that the spatial gradients are as much produced by inactivation mechanisms as rate of diffusion. So we said, let's eliminate this binding site. If we remove this inactivation mechanism, the gradient should go away. And I lost a beer on that one. So the threonine 75 is not acting as an inactivation mechanism. It's really a diffusible buffer. And we're still exploring what would be required, what sort of mechanisms could produce a pKa gradient. We have a collaborator who is looking at whether there are indeed gradients of pKa in hippocampal neurons, but we don't have results there yet. Okay, well, I've just shown you a lot of simulations where all we're doing is activating the dopamine receptors, and I don't have any calcium influx. But of course, calcium is required for LTP in the striatum. Here shown experimentally, this is our control high-frequency stimulation. If we add AP5, which blocks the NMDA receptor, we see no LTP. Well, a previous model that Jeanette and I collaborated on, it was a single compartment model, and it shows that this is the pKa activity with dopamine alone, and then when you add calcium, you see this facilitation. Well, how is calcium causing this facilitation? Indeed, calcium inhibits adenylate cyclase 5. When it binds to calmodulin, the calmodulin can then bind to protein phosphatase 2B, which actually dephosphorylates DOCK32. It can activate a phosphodiesterase, which inhibits cyclic AMP. So we have many pathways whereby calcium actually inhibits the activity of pKa. Well, there's one pathway where calcium can actually enhance pKa activity. Calcium binds to and activates or further activates protein phosphatase 2A, which then dephosphorylates threonine 75, which releases inhibition of pKa. And in this model, this was a very strong excitation. This pathway the rate constants had not been measured. But after we did our study, those rates were measured, and it turned out that this is a very weak excitation. So in our new model, here's the effect of dopamine on our pKa. Here's dopamine plus calcium. We see a weak inhibition. We do not see facilitation. So we still don't know what the calcium is doing. Well, one possibility is activating CAM kinase. And indeed, when you add the CAM kinase activity goes way up, not surprisingly. So one possibility is that you need not just pKa for plasticity, but you need both pKa and CAM kinase. And when you give both dopamine and calcium, the sum of these is quite a lot more than either one alone. Well, it's very difficult, but by the way, the evidence that CAM kinase is important in the striatum is rather tenuous, and I'm not sure whether that's because people just haven't looked at it that much or whether it's not actually involved. But from a modeling perspective, what we really need to be able to do is compare two different temporal patterns. A temporal pattern which produces LTP versus a temporal pattern that produces LTD, and then we want to know which molecules are activated by calcium more strongly with our LTP protocol and which ones are activated more strongly with our LTD protocol. Now, in the hippocampus, that's pretty straightforward because high frequency gives you potentiation and low frequency gives you depression. But in most cases in the striatum, high frequency produces LTD, and you have to remove magnesium from the bath to get LTP. And so that's not going to be a really nice paradigm for us. So instead, my lab developed a theta burst protocol that would give us LTP. And the theta burst protocol we used was at basically 10.5 hertz between these bursts, and we gave 4 pulses at 50 hertz. And this produces a very nice LTP shown right here. We're measuring this using field recording. So here's the pop spike prior to our induction protocol. Here's the pop spike afterwards. We're measuring the peak of the pop spike. And this shows our theta burst protocol. This is with no stimulation, so it's not just a time-dependent effect. And then this is indeed NMDA dependent. And we can get this not just using field recordings. We can also get this when we use whole cell recordings. And interestingly, 100 hertz does not give us this long-lasting LTP. 8.5 hertz does give us, so we're now looking at various protocols because we want to make sure we're using the best one. So now we can use these 2 different stimulation protocols, stimulate the model with both calcium and dopamine, and ask which molecules are discriminating our temporal patterns. And so what we see here is that theta burst stimulation actually is not the best way to activate CAM kinase. Instead, we get more CAM kinase with our 100 hertz protocol. And those of you who know a lot about CAM kinase, this is not all that surprising, but it argues against CAM kinase being implicated in our theta burst LTP, because that, if it were the case, we should get 100 hertz should produce a nice, robust LTP. So now we looked at our PKA activity, and indeed it's roughly twice as large the activity if you look at the total area under the curve compared with our 100 hertz. Protein phosphatase 2B has the opposite effect. We see it is indeed lower in theta, which again works in our favor, but this is a 2-fold increase. Is that really the only molecule? And it's not necessarily due to the calcium. So again, we're looking for calcium-activated molecules. Well, one of the things that we didn't have in this first model are GQ-coupled pathways. It turns out that for LTD in the striatum, we need production of endocannabinoids. And that requires GQ protein activation, which binds to phospholipase C and produces diacylglycerol. Well, that pathway leads to 2 different molecules, the endocannabinoid called 2AG, which is implicated in LTD, and also protein kinase C, which is implicated in LTP. So we created this other model, and right now I'm just going to tell you results just using this part, and we haven't merged them yet. And we're going to stimulate with calcium and metabotropic glutamate receptor activation using our 2 different temporal patterns. But first we're going to validate this model using a different protocol called depolarization-induced suppression of inhibition. And so these are our redrawing of the experiments. If they give either 0.1 second, 1 second, or 5 seconds of depolarization, and on top of that they add various amounts of the metabotropic glutamate receptor agonist, what you can see is different amounts of suppression. But basically, the larger the mGlu-R agonist, the larger the suppression, as long as you're only giving short depolarizations. But once you give this long 5-second depolarization, which probably brings in a lot of calcium, then the DHPG doesn't help. And so in our model, we compared our results by looking at 2AG, either with calcium alone, which would be consistent with just the depolarization, or in the presence of DHPG. And you can see that we get similar results. So this made us very happy. We also wanted to validate our model by looking at PKC translocation. And so what you have here is the holoenzyme, the calcium-bound form, and the diacylglycerol-bound form. And the diacylglycerol stays in the membrane. So now our calcium is coming in. So PKC goes up everywhere, but it binds to the diacylglycerol, and once it binds, more PKC diffuses to the membrane and binds further. So what you see here is translocation with the temporal, with the dynamics similar to what's been measured using various GFP constructs. And, sorry, it does this. But we do not need any sort of active translocation mechanism. So just binding to a membrane-bound molecule and free diffusion is enough to explain the observed translocation. So now we took this model and we stimulated with our theta burst and our high-frequency stimulation, and we looked at the 2AG, and the 2AG activity only increases about twofold when we use theta, but now the PKC activity increases greatly using theta bursts. So, and this is just summarizing the total amount of molecules, and here's the increase. Here's high-frequency stimulation, slight increase in 2AG, and the high-frequency, a larger increase. So we think that probably protein kinase C is involved in the theta burst LTP. And we don't necessarily think that any single molecule is involved. So the enhancement of PKA with our theta burst clearly can be involved in LTP also, and we're planning to experimentally test the role of PKC using some PKC inhibitors. And now I just had to have my own omics on here because that's not necessarily the whole story. We know that there are many signaling cascades in these neurons, and so Jeanette is currently developing new methods to try and identify other molecules that we need to include in our model. We certainly don't want to put in every molecule that's around. It's just not computationally feasible. So we can say go to the genes to cognition and get a list of proteins, which many of them are in the PSD, some of them are probably contaminants. Then we can take those list of proteins and use them, say, as bait to go into these protein-protein interaction networks to extract these interaction partners. The problem here is that it's not cell-specific. And again, the problem here, these are whole brain samples. We want to know what's in these spiny projection neurons. So now we can go to these transcriptome databases and try and identify cell-specific interaction partners. And so this is just an example where we use PP1, protein phosphatase 1, as bait. And here's the filtering. And so here's a list of molecules, but they're not all in spiny projection neurons. But here's a set which are related to PKC. Here's DARK32 and inhibitor 1. And so what we're really going to do now is try and find some of these other calcium-binding proteins that might be involved, especially if our experiments blocking PKC turn out to be negative. All right, I'm going to skip this part in the sake of time, and I just want to thank all the people that did this. I just started a tradition in our lab. When we get a paper accepted, we go to ice cream, and so this is, Bohan Kim did the work with 2AG, also helped by Lane Wallace. Rebecca's done the whole cell recordings. Sarah Hawes did the field recordings. Wanyul is working on the software, and those that came to my poster, this is Sriram, who is not here because of a broken ankle, but he'd love to be here. And of course my collaborators, Dietmar Plenz and Jeanette Helgen Kotelewski. Rodrigo did the PKA simulations. And thank you again to the funding agencies.